Ethylene is best known as the hormone in plants that tells fruit when it's time to ripen. It's what tells a green banana to turn yellow or a tough avocado to soften. In plants, the effects of ethylene have been observed for decades, but little is known about the effects of ethylene on bacteria. In the lab, I worked with a bacteria called Bacter xylanus, or KX for short. KX produces very large amounts of cellulose, which is the same tough material that gives plants their structure. In bacteria like KX, cellulose helps form biofilms, a city of microbes that adhere to a surface. But what determines how much cellulose this bacteria produces? This is where ethylene plays a role. Ethylene, the gas released by plants, can be in close contact with KX because KX lives on plants. Our lab has already shown that ethylene increases cellulose production in KX. The real question is, how does KX actually sense ethylene, and how does it control cellulose production in that bacteria? This summer, I studied a not yet characterized protein found in Bacter xylanus called ESH, which we hypothesize acts as an ethylene sensor. Think of ESH acting as a potential ear for the bacteria, allowing KX to detect plant signals like ethylene and trigger vital biochemical pathways like cellulose production. To pinpoint what exactly ESH does, I used two main approaches. The first method was bioinformatics. With the help of computer tools, I was able to compare the ESH protein sequence to other known proteins. Results showed that ESH is very similar to histidine kinases, which are proteins involved in signal detection and response, implying that ESH plays an integral role in signaling pathways. I also found that ESH is located in the cytoplasm, which makes sense, seeing as how ethylene is a gas and can easily diffuse into the cell. The second method was genetic engineering. Within the lab, I am currently working towards removing the ESH gene from KX. Why? Well, if ESH really is the sensor that allows the bacteria to hear ethylene, deleting it should be like plugging in the bacteria's ears. This would allow us to test whether or not the ESH gene is responsible for ethylene detection in KX. If ESH really is the ethylene sensor, the implications of this would be very far-reaching. What if we can utilize the bacteria to detect stress signals from plants to prevent spoilage or even trigger on-demand biofilm production? What if we could use the fact that bacteria responds to ethylene to produce more plant-based materials like cellulose and biodegradable plastics that would help our environment? So although my project focused on studying how one protein listens to a tiny molecular message, the discoveries have the potential to revolutionize agriculture, biotechnology, and environmental sustainability.